If you're preparing for biology paper 2, here is one of the most important required practical, investigating the effect of temperature on the rate of decay of milk by measuring the time it takes for the pH to change. Before we go through this method, there is one important thing I would like you all to know. Whenever you are using lipase, remember that lipase is an enzyme, and lipase breaks down lipids into glycerol and fatty acid. This is the fundamental idea behind this practical. So I'm going to write down at the top. Lipase breaks down the lipids present in the milk. Remember, the milk contains fats or lipids. These are what the lipase act on. Okay, so let's go through the method. First, measure 5 centimeter cube of lipase solution using a measuring cylinder. Remember, always write the equipment you are using in your method. Then place the lipase solution into a test tube. Next, measure 5 centimeter cube of milk into a different test tube and 5 drops of phenolphthalein indicator and 2 centimeter cube of sodium carbonate solution. Now remember and listen carefully, sodium carbonate is an alkali. The color of phenolphthalein in an alkali will be pink. This is exactly what we want. We want this milk mixture to be pink and in alkaline condition so that we can observe the color change clearly in the next stage of the practical. What we do next is place both test tube in a water bath at 30 degrees Celsius and leave them there until they reach the same temperature. Students are often asked this in an exam. Why do we place both test tubes in water bath before mixing? And the simple answer is to ensure that both solution reaches the same temperature before the reaction starts. Once both test tubes have reached at 30 degrees Celsius, quickly use a pipette to add 1 centimeter cube of lipase solution to the milk test tube. Remember this, the milk test tube that is pink because it contains phenolphthalein and sodium carbonate. As soon as you add lipase solution, start stopwatch immediately. Then stir the content using a glass rod. As soon as the solution loses the pink color, it becomes colorless. Stop the stopwatch, record the time in a result table. Now, why has the color changed? This is another very important question. The color changes because lipase has broken down lipids in the milk into fatty acid. The fatty acid lower the pH of the mixture. Earlier, the mixture was alkaline because of the sodium carbonate, and phenolphthalein is pink in alkaline condition. As more fatty acids are produced, the fat pH falls. The mixture is no longer alkaline and the phenolphthalein changes color from pink to colorless. The time taken for this color change is what we measure. Now we look at the variables. Remember, the independent variable is what you change, and in this practical, the independent variable is the temperature of the water bath, because you repeat the experiment at different temperature. The dependent variable is what you measure. In this practical, it is the time for the pink color to disappear and become colorless. Control variables are very important. These include volume of lipase, volume of milk, number of drops of phenolphthalein, volume of sodium carbonate solution, stirring method, type and size of the test tube, amount of mixing. There are many control variables you could mention, but make sure you include them some in your answers because examiners are looking for them. Another important point, if you are repeating the experiment at different temperature, keep stirring method the same each time. For example, 5 stir at one temperature and 5 stir 5 times at every other temperature as well. Students often forget this. Another very important point is to repeat the experiment 3 times at each temperature and calculate a mean. If an exam asks how reliability can be improved, a good answer is to repeat the experiment 3 times at each temperature and calculate the mean. This is very common mark scheme answer. I have also included additional common mistakes and exam tips on the revision sheet attached below. You can download the sheet using the link below, and thank you. Do let me know if you have any question. Thank you.